Shaking up DNA, a dance of genomic proportions. Picture this, a high-stakes game of genetic musical chairs played right inside your cells. Sounds crazy? Well, on January 31, 2025, a team of audacious researchers led by Jonas Koeppel from the prestigious Wellcome Sanger Institute published their ingenious work in the powerhouse journal Science, and they've given new meaning to the phrase gene shuffle. In their groundbreaking study, Koeppel and colleagues tackle a long-standing puzzle, the vast sea of non-coding DNA swimming in our cells, much of which seems to be just loitering about, doing seemingly nothing. But don't be fooled. This is no slackers club. Non-coding DNA might just hold the key to some major biological secrets. These bits and bobs have eluded the tight focus of genome engineering because they haven't managed to wiggle into the spotlight as well as their protein-coding cousins, which only make up about a measly 1% of our genome. So how did the scientists plan to unravel non-coding DNA's mysterious dance? Through a jiving new technique called prime editing, paired with their trusty sidekick, CRE recombinase. Here's how it works. The researchers ingeniously inserted CRE recombinase sites into repeated DNA sequences using prime editing, a tool that makes the genome itself more flexible than a cat in yoga class. This move essentially laid out a genetic dance floor, allowing for all sorts of recombination hijinks. Once the floor was set, Koeppel's team unleashed CRE recombinase to orchestrate a genomic shuffle-off, producing massive rearrangements across the entire genome. Amazingly, they generated more variations in a single cell than a universe of dots in a Pointillist painting, think tens of thousands. After the party, they monitored which cells managed to stay groovin'. Cells shuffled in such a way that they either hit a lucky streak or bailed out of the evolutionary line dance. This allowed the researchers to pinpoint essential gene players needed for basic cellular dances and moves that might stir a future in biotech innovation. It turns out, the genome is a master at improvisation. While many cells toned down their tunes due to rearrangements that rendered them unfit, those that survived offered a treasure trove of insights. This not only painted a picture of gene tolerances and non-essential sequences, but also revealed non-coding dancers that groove to their own mysterious beats. Though they've made killer leaps, our ambitious ballroom dancers admit the road isn't without hurdles. Managing this colossal array of genomic variations was like conducting an orchestra of cats. Non-transformed cells are particularly finicky dancers, reacting to the shuffle with a hissy fit that involves DNA repair madness. Yet, the opportunities arising from understanding genome organization are staggering. This is just the opening ceremony towards defining a minimal genome tailored for optimized performance, a dream both scientists and synthetic biologists alike have harbored secretly while daydreaming during their downtime. Looking ahead, the implications stretch far beyond simple curiosity. This scientific twist could redefine how we craft cells for biotechnological uses, such as drug production or understanding the mechanics of drug resistance. Who knows? The next few decades might involve synthesizing custom genomes for everything from better yeasts for brewing to dye for crops made more resilient against pests. In the grand spectacle of discovery, Koeppel and his team have given us a masterclass in genetic dance composition. As researchers around the world study their technique, we find ourselves not at the end, but just at the beginning of an epic genomic saga. So hold on to your pairs of alleles, folks. This genomic concert might just outperform Beethoven's Fifth.